neuroscience and beyond. Know more, get inspired. I'm Svilen Georgiev, and I'm a PhD student at the University Medical Center Göttingen in Germany. Together with my PhD colleagues Sabina, Christina, and Lenny, we desire to bring science, and especially neuroscience, closer to the general public. In the following episodes, we're going to talk to various scientists about their inspiration how to do science, about their research topics, but also about their opinion on current scientific problems. Today, my guest is Dr. Felipe Opazo. Dr. Opazo is a principal investigator at the Center for Biostructural Imaging of Neurodegeneration in Göttingen. His group focuses on finding molecular tools to study the nanoanatomy of neurons and other biological systems. Dr. Opazo and his team develop tailor-made small probes that are able to detect and quantify molecules in living and in fixed cells. In addition to his current research position, Dr. Opazo has co-founded a company called NanoTag, which mainly produces single-domain antibodies, also known as nanobodies. Before we begin, I would like to mention that this podcast is separate from my research position at the University Medical Center Göttingen. This podcast is supported by the International Max Planck Research School for Neurosciences, as well as the Cluster of Excellence Multiscale Bioimaging in Göttingen. And now, please enjoy the talk with Dr. Felipe Opazo. Hello, Felipe. I'm glad to see you here now, and we are very happy to have you. And also, I'm happy that you actually came despite the storm outside, snowing. How are you doing today?
Thank you very much for the invitation. Yes, great to be here as well.
Can you please tell the audience, we know each other for roughly 1 year since we are colleagues in the department, but could you please tell us more about yourself and how did you end up in science?
So from my origins, I'm a national. I was born in Argentina, let's say, but grew up in Chile, and then I came back to Germany when I was already around 23, 24 years old. And this was coming here to do science, exactly. So my interest from science was, I don't know, I cannot explain it. My family, there is no scientist. But I had the interest already at the school level, and I decided to go into a scientific career in Chile back then. And then, of course, I was motivated to do my PhD in Europe, and I ended up here in Göttingen.
So what was actually your undergrad study in Chile?
So I wanted to study biochemistry, but my family didn't understand what it actually was, and they freak out that they say, no, we're making an effort for you to go to university. You need to study something that makes sense, that like law or medicine or something like that. And I said, no, I want to study biochemistry. And back then appeared a new career that it was had a fancy name, a very pompous name. It's called molecular biotechnology. And I said, look, this is related with business and science, and somehow they accepted, and I managed to study there. It basically was biochemistry with a little bit of business and math, but it basically was in the biochemistry faculty. But so this is how I got around to be able to study what I wanted.
Oh, that sounds great, actually. But when did you find out about your interest in neuroscience? Was it because of this program that you applied later on?
No, it was already there. So when I did my bachelor in Chile, I was influenced by some of the professors or teachers there that they were very much into the neuroscience. So the Faculty of Science there in the University of Chile was quite directed to neurosciences. And neuroscience is one of the fields in Chile that is, I would say, relatively strong. Relative, because in Chile there is not a very strong science, but I think there are some good, contributions to the neuroscience field coming from Chile. And this was the interest. So I did my bachelor thesis or work there already related to Alzheimer and neurodegenerative diseases back then there. And then, of course, I was looking for programs in Europe. Part of my bachelor thesis or my master's thesis, which I did there, I came to EMBL here, to EMBL in Heidelberg, and I was blown away looking at how things are running here compared to the a more much poorer country. And So I said I have to come back to Germany or to EMBL. In EMBL was complicated because as a Chilean national, you actually cannot. It's only dedicated mainly to European, at least back then. I don't know if this changed. And then I was looking alternatives, and there were 2 Max Planck programs for the IMPRS, right? And it was the neuroscience, the molecular biology as well here, but it was also applied to something in Dresden. Right? It was also in the developmental biology, I believe. I applied to the neuroscience and developmental biology, and I liked it much more than the neuroscience, so I decided to come here at the end. So that's why I ended up.
That is great. And the people who don't know this, actually, the IMPRS stands for International Max Planck for Neuroscience.
Sure.
And I'm also a part of this program. Felipe is one of the first batches. I don't know how many years ago. I don't want to tell you.
I was the second or the third batch of the PhD students.
Yeah, but what was the program for you? What did the program give you, actually? It's a very nice setup of people who gather together from all around the world, roughly 20 people. And you're put into a one room for roughly 1 year, where experience a new country, new culture, and also work hard to study and take your exams and eventually continue in this scientific career. What was this program for you?
I love it. I mean, back then it was probably much less organized than how it is now. I think today the organization level is much better. So as I said, I was, I think, the second or the third batch, but it still was an amazing experience. So this was coming to Europe, to start this adventure, and everything felt so well organized, although it was probably not as well organized as today, but for me it felt already quite amazingly organized, and you were having a little budget, we're able to have a room, and actually this was, I like it very much. This year was a fantastic year, also I guess because the internationality of it. So I was put together with people which I'm still very friends with, all of course all in the program, and we were in a VG, in a house where 4 or 5 now, 4 I think, or 4, and each one from different nationalities, and you know in Chile there is not a lot of multicultural, so it's very far from the rest of the world, and it's hard to see people much immigrants or people coming to Chile if it's not tourism. So it was for me a very, very enriching experience of exchanging and sharing. I remember this time with very cool, very, very nice actually. It was pretty nice.
Yeah, for me it was also nice, but unfortunately I had this bad luck that Corona happened, and in our batch, this program is known to this cultural events and all the social part. You go to see the labs and the PIs and PhDs, and we actually had to stay home and think about our science alone and at home, which was a little bit depressing, but hey, however, we made it to go outside to meet a little bit, have fun, and now I'm still here, and eventually also you stayed here. What happened after graduating from our program?
When I finished my PhD, also in the neuroscience field, in the neurodegeneration, so I was also working related to Parkinson's disease in my PhD. So I moved from my master's thesis in Chile that was Alzheimer, then I've been to Parkinson, but I kept, of course, in the neurodegeneration. There were several things to stay here. They, at that time, was very, very interested in microscopy, and in Göttingen there were happening one of the strongest revolutions that one could think of in terms of microscopy. So I decided that it was a little bit silly to leave the place where actually the super resolution effervescence was coming. I mean, it's not the only place, but was definitely one of the big places where you would like to be if you want to do microscopy in neuroscience. And this was, I would say, one of the main reasons. The second reason was, of course, I got a family here, and actually it was convenient to also stay. We could have moved. I think my wife was back then ready to actually also take the adventure and I don't know, go somewhere else. But as I said, it was also convenient to stay. But the reason was the microscopy, the science here happening in Göttingen was very strong. It's still very strong, actually.
So from neurodegenerative diseases, you found your love for microscopy somehow.
Yes.
And also method development, as we can hear maybe later when you tell us more about your current position and current projects.
Right.
But how come that you switched? Is there any trigger, or you just found some, let's say the STET or some other microscope? STET is a super resolution microscopy technique, which we'll hear more about it a little bit later. And what was this trigger for you personally to switch from these two fields? Because they're kind of different. You deal with disease, and on the other hand, you're more to the method development side.
So I was doing, my PhD was basically measuring amperometric recordings and electrophysiological recordings on dopaminergic neurons, and which is fine. It's a technique that is very, very informative, very precise. But at some point at the end of my PhD, and this was my own side project, I decided to follow one of the molecular phenomena by microscopy, and I started doing time lapse. In the lab where I was at back then, they didn't have very microscopy or equipment, so very, very special microscopy equipment. So we need to always go to a neighbor lab to get some confocal time and try to do some time lapse. So, but it came from my incentive or my idea to look at things, to look at the processes, not only measuring them, which is again very different to what I study. So I'm basically by training a biochemist, but then I moved to electrophysiology and suddenly I started moving. I don't know, this has happening to me constantly, that I move fields. I don't know, I wouldn't say I get bored of something, but I get curious to get to try something else at some point. And this was at the end of my PhD. I did a lot of microscopy, live microscopy and time lapses with conventional epifluorescent or confocal, so nothing really special. But then is when I got to learn all these new technologies coming from Göttingen and also somewhere else, right? So I decided, yes, I want to maintain the neuroscience focus, but based now on microscopy. Let's see microscopy. And then, as we will probably discuss, I will change once again a little bit the gears and maybe going back to my origins of biochemistry. But yeah.
Yeah, this sounds very interesting, and actually this makes you a specialist in different domains, or not a specialist, but at least you know many perspectives, and you can combine them and use the synergy of these many techniques, perhaps, future research. And by the way, just to mention, for everyone who is not familiar with these terms that we use here, like confocal microscopy, fluorescence microscopy, we will try to explain it in the description and give you more information. So, but now you went from biochemistry to electrophysiology, and now you're doing, developing things for which will help the microscopy technique to be even more precise than we have it now. What are you doing actually in your current job, so to say?
So, again, to put them a bit in perspective, in 2012 we have a manuscript using super resolution microscopy, so a microscopy that can go beyond the diffraction limit of light, which back then was a big thing. Today is becoming more like a standard thing. But in 2010, this was really relatively new. And then already we had a manuscript where we basically observe that if you make your staining or you label your sample in a more intelligent fashion that has been done before, you can extract more information from the super resolution microscopy. This was also not an issue or not a problem before, because there was not the need of having a more precise labeling, since the microscopy was anyway limited by its diffraction. This was with aptamers, which are small DNA or RNA molecules, but the key point is that they're small. And the classical way of doing it is with antibodies. You will have an antibody against your protein of interest, which you will either directly put a fluorophore or recognize with another antibody recognizing this antibody, which is used to amplify the signal. And that will typically result in an immunostaining or a labeling of your protein of interest. So this antibody complex, primary, secondary antibodies, typically finish in 20 to 30 nanometer resolution, which back then was not a big problem. Still, super resolution was in the 60 nanometer reality, which now is indeed a problem. We are in the one digit nanometer resolution, and this is I think this was a good decision to move into improving the labeling technique to study my neuroscience questions. So that was the point. I thought at that point we were looking at a lot of images with STED microscopy, which is a super resolution technique, using antibodies. And the conclusions were limited because the way of labeling. And we had this seminal paper where we showed that if you reduce the size of your probe, the labeling gets much better and you start seeing things that you were not seeing before. And this was the trick to say, okay, before I really answer the questions I have in neuroscience, I need to improve my way of labeling them. And this was, it was also a very good excuse to come back to biochemistry, I would say, because I have done all this path, and I didn't mention, but when I was in EMBL, I was doing molecular dynamics. So it was all computers and playing with molecular docking and getting in Alzheimer's. So I think I did a big roundabout in order to get to biochemistry once again, making small probes that they will serve, they will deliver a better tool for basically for neuroscience and any other field in this super resolution new era, I would say.
That's great, and it sounds amazing that you actually came up with this idea and you guys found out about these things. But can we go back a little bit and slow the gears a little bit and explain actually to the people who are listening and are not familiar with these terms, what are actually antibodies? Maybe people heard about it quite often during Corona, especially in the US. They had this monoclonal antibody treatment.
Yes.
What are these monoclonal or polyclonal antibodies, and what is actually antibody, and why is it this size so crucial, as you already mentioned, and how can you overcome afterwards this labeling mistakes, so to say, because of the size of the antibody?
So very, very generally speaking, antibodies is a great invention from nature, where is what we call our immune system, part of the immune system, not the full immune system, but part of our immune system is generating antibodies against pathogens. And these are relatively large molecules because it has many functions. But one of the important function is to recognize something in a very specific fashion. So in order to bind a virus, a bacteria, a foreigner agent that is in your body, that will be the principal aim of antibodies. And upon binding, of course, they will call the whole army to come, leukocytes and whatnot, to come and try to help to eliminate that threat. So antibodies has been evolved as long as mammals or has been around, basically before actually, and exactly birds, they also have antibodies. So it's much old. But indeed, science came with an amazing way of recognizing specific molecules, and this is really at molecular level. So we have the capacity in our bodies to generate antibodies, and these antibodies are relatively large molecules in comparison to a protein. It are basically, I don't know if I should go to the specifics of how do they look like, but they are a tetra. Basically, they are a dimer of two similar chains, heavy chain, light chain. They are relatively large. They are 150 kilodalton, which is a very considerable large protein. Because they have two domains binding their target or epitope, to be correct, and they have this Fc domain, which is typically used for the dendritic cells or other type of immune cells to recognize that there is an antibody floating in your serum or your plasma in your blood. So science very early on used these tools to use them to recognize specific proteins. So you will immunize a rabbit with a particular protein X. This rabbit will generate an antibody against protein X, and you can extract a little bit of blood of the rabbit and then use this serum or these antibodies that the rabbit generated to detect particularly protein X. So this has been a tool that has been used already, I don't know, from the 1800s. From that region more or less, come from that time. That has been used. So this has been science doing it a lot, and in order to detect in fluorescent microscopy, in order to detect a particular protein, has been typically use antibodies. And you have antibodies against insulin, antibodies against tubulin, antibodies against classical proteins, where you will be able to detect and reveal your protein of interest. So that is the classical way of making an immunolabeling, immuno from antibody labeling. As I said, in terms of size, they're 150 kilodalton in weight, but in mass, but they actually around 15, 20 nanometers. They are very flexible, so they might change a little bit their size. But if you go linearly, they can be up to 20 nanometers in length. Mm-hmm. And if you now need to put on this antibody another antibody where the fluorophore is that your machine or your device or your camera will detect, you're having this what we were discussing, this linkage error. So between your target and the fluorophore that you are detecting. At the end of the day, you're only looking at the fluorophores. You're not looking at your protein of interest. You look at the fluorophore. But you expect this to be close enough that you don't make a mistake. And this with conventional microscopy, this was not a problem ever, because the resolution of a conventional microscope, a very good one, will achieve, 200 nanometers. So adding this 20 nanometers to 30 nanometers error, it wouldn't have a great influence. But now that we are able to look at 5 nanometer resolution, putting your fluorophore 30 nanometers apart is indeed a problem. So, as I said, we observed this problem in 2010. This became a paper in 2012, and since then I've been putting a lot of effort in making new probes and sensors.
Okay, I think people will understand now after this explanation what are antibodies, why do we use it in science, in particular in neuroscience. We label molecules in the brain very specifically, and these antibodies are attached, or to these antibodies we have another compound, so to say, that emit that fluoresce and emit photons, so to say, and we can detect them. However, you said now you use these aptamers, which are smaller than antibodies, but you also use now the so-called nanobodies. What are these things actually?
Nanobodies, it's another type of antibodies that again, evolution or nature did something which we don't know why, but in so far we know 2 species of animals that they make a slightly variation of the classical antibodies, and these are the sharks and the camelids. So camels, dromedaries, llamas, alpacas, the new world camels, as we know. So these 2 animals, and it's not quite clear. These evolve completely independently, obviously, because they are very different in their in their origins. But these antibodies are slightly smaller than the conventional antibodies. And important, in order to recognize their target, they need only a single chain of protein. While in normal antibody, didn't explain that, but you typically need 2 chains to be in a very specific position in order to recognize your target. And this was a serendipitous discovery in Belgium in the early 90s, where they were examining blood of camels, and they recognized that there was a size of an antibody that didn't match to the size of the classical antibodies that everyone thought that every mammal will have. And these were, then they found out that these were smaller. And they coined this nano because they are much smaller. The antibody in the animal is slightly, it has an Fc domain, as they were saying, and it's a dimer. But since it's a single chain protein, one could find out the sequence and produce only the relevant part, and this become really 10 times smaller than a conventional antibody. So it's in the 15 kilodalton mass and in the range of 3 nanometers size. So it is considered much smaller.
Do we know actually why these guys have nanobodies and these animals? Do they have actually antibodies also? Do you know
They
that?
have conventional antibodies and another set of these antibodies, which is, from the immunological perspective, is very interesting because they need to have a completely two set of B cells, and the whole rearrangement of these V, D, G, J sequences is actually not very well studied. I think there are some clues of how it happened, but is not as clear as with conventional. But actually, I don't know if sharks will have conventional antibodies. I think they don't. They have only these type of antibodies, while camels have both. Have the conventional and these were called heavy chain antibodies because they don't have a light chain. Mm-hmm.
Okay, that's interesting. Maybe we can check it later if sharks actually have also this. But okay, how long would it take you to inject, in this case, a llama or alpaca, and then try to produce, so to say, a nanobody? And is there any problem? So you mentioned they are smaller and you have to attach this fluorescent molecule to them. Is there a problem in comparison to a conventional antibody in terms of the fluorophores that are attached?
So in terms of time, it takes an immunization campaign. So you will, if you will want to have alpacas or llamas making nanobodies against target X, protein X, you will need to immunize these animals, so vaccinate those animals we do around 4 months of vaccinations, and they get a vaccination every 3 weeks or something like this. And after these 4 months, you will take 100 milliliters of blood, and then you will start the analysis. Of course, in the blood, you will contain all the antibodies these animals generate, or at least a big proportion of them. And then start the screening, the search for which is the one that is binding the target I want this to have. And this is a whole biochemistry process. It's basically based on phage display, which was recently recognized as a very important technique with a Nobel Prize.
Can you elaborate a little bit about this phage display?
So this is using, again, another nature trick, which is this gigantic war between bacteria and viruses. This has been very early on happening. Bacteria and viruses have been fighting for really millennia, and of course they are these bacteriophages, and these are viruses that they specialize themselves in basically in attacking bacteria. And bacteria, of course, have developed in these millions of years tools to actually counter-attack the viruses. But we use these viruses because they are very interesting, these bacteriophages. In short, we call them phages. That's what is phage display. And we get the whole diversity of nanobodies that, for example, a llama in a particular time is making, and we make a library of all these constructs. So we basically extract from this 100 milliliter of blood, we can extract all the genetic information that this llama is producing in terms of nanobodies, and we clone them into a phage. So we make now these bacteriophages displaying a particular nanobody, and each phage will display in its surface a single nanobody. This is very interesting because the phage contains inside itself the genetic material that it produced the phages. So it will also contain the sequence of the nanobody that is in that moment being displayed by this phage. And what you need to do at the end with the phage display is you make a soup of the whole diversity, which is in the range of a couple of million different nanobodies that we put in viruses. And then we use these viruses to basically get immobilized by having the antigen in a resin or in a solid support. And then you wash everything that is irrelevant, and then basically you can elute those phages, and you can send or basically get the sequence of the nanobody that is being displayed in the front. So the phages they are really attacking the bacteria, but we use them as a tool to link the protein with the DNA sequence that code for that protein. That's the phage tool, basically.
This is just amazing how we can utilize things that are happening normally in nature and use it for your own purpose to, in this case, produce these guys called nanobodies. And I mean, I've been always astonished by science, but this microscopy, as you, I also find it more than great that you can actually see things, in our case, in the brain. And nanobodies, I also use them a lot in my research, as you already know. And I'm very happy that we have these tools and we're able to study the molecular machineries of brains and synapses in particular. And synapses, for people who are not aware of, are contact points between two nerve cells. And we might explain this through our talk today. But besides, so you are working as a PI, principal investigator, in Göttingen, Germany, where are we now located? And you also are involved in another activity, and this is a company that you actually co-founded a couple of years ago.
Yes.
Could you please tell us more about this nice adventure?
So again, I would say this comes back to the time where we had these aptamer papers where we realized that the size actually really makes a difference for super resolution microscopy. And at that point, it was, at least in my mind, I don't know if it was for everyone like this, but I definitely saw that this trend of using super resolution is going to only increase. So people will at some point, if this become a technique that is so ubiquitous and everyone is using it, then everyone will need another way of labeling their target, their protein of interest, or the subject of study. And then when I said, okay, I think we need to move into maybe provide this support. So now it's very easy for most of scientists don't need to make their antibodies. You just basically purchase them. You just go and buy an antibody against your protein of interest, because the catalog is gigantic. It's still not big enough. There are a lot of proteins that don't get attention because they don't have an antibody against this. But the catalog of antibodies is relatively large. However, the catalog of small probes, it was back then reduced. Now it's slightly larger, but it's still not even a 1% of what you will encounter in the antibody catalog. So really the original intention was to generate a company doing R&D, research and development, to generate probes for doing better science in the field of super resolution and biochemistry at the end as well. But this was the thought. So we thought we were at the point we realized something new. Small probes are definitely going to be needed in the future. And this was the point where I decided, okay, we need to try to make a company generating this.
So you said we need to try to make a company, but I guess making a company is not an easy thing, and it took time. And could you please tell us more about this process from the time that you have the idea in your head until the final setting up of a company, for example?
So the idea, this was again 2010, 2012, in that region where I was working in that manuscript. It was a very difficult manuscript to put out, because we were telling the community that antibodies were not doing a good job, and people didn't want to hear that. And actually, it was very difficult to get the manuscript to get accepted. It was more than a year and a half back and forth in journals. And typically the criticism is like, we don't believe that the antibodies are so bad. So it took quite a bit. Now it's quite accepted, and everyone talk about this linkage error, and of course it's a problem, but back then it was very hard to hammer it into somebody to believe us. And so this is where I started, 2000, let's say 11, 12, and the company was founded in 2015. So you can think that it took 3 years, and this was, of course, I spent quite some time trying to make a business plan, business model, to see if this is going to be sustainable as a company, which I receive a lot of no, it's not going to be sustainable as a company, because I was aiming to generate reagents for research. So I was not doing to cure cancer or do a therapeutic something. And this is still the belief. So it's very hard to keep a company alive if you generate only reagents for research. It seems to be not a big pot of money, let's put it that way. So investors are not very keen in investing in something that will bring not a lot of money. And this was indeed complicated to find somebody believing that this is going to be necessary for the research field. And on the other hand, you don't get governmental support to do such as project because it's even considered sometimes not very scientific. It's a technical work. You need to generate probes. Although in my view, this is vital because without this, you cannot move forward in the other context. But that's another discussion. So indeed, was not easy. We knocked several doors. As I said, it was a 3 years effort. And at the end, like everything, you need a little bit of luck. And we got in contact to a company that is local in Göttingen, that is one of the famous antibody companies for neuroscience field, Synaptic Systems, and they believed in us and they gave us support to start to initiate this adventure. So in that sense, one need to also be lucky. As I said, we knocked many doors. At the end, it was very close by. It was in Göttingen where we found at the end the investment capital.
So take-home message networking is very important also?
It's vital. Actually, you should start doing networking yesterday because it's exactly where things can move and get further, yes.
Yeah, and here we are now in 2022 and the company is still there and is successfully running and even more successfully than expected, as you mentioned previously in our private discussions. But I would like to go back again from this starting point. You were there working scientist, I guess a postdoc at that time, and you managed to find time and invest this time into creating a project. How do you manage to do this? And also, if I may add another question, did you have your family at that time? And this is also a thing that scientists always have to think about their work-life balance, because our job is filled with activities that we have to fulfill on short deadlines, and also on different aspects that we have to do. For example, having a project, thinking about the theory behind it, the practice, designing experiments, also gathering data, analyzing this data, writing your paper, writing grants, etc. So this is a huge effort from each scientist, and I'm very, very keen on understanding more about your approach to this, so to say, time management during that time, for example.
Yeah, I don't know if I have a direct answer to you. It kind of grew with the need. What I realize now retrospectively is that how much time I had back then, right? So I think it's very hard to get out of your routine. You have the routine and you think that you are full with things and you cannot fit anything else in your timeline. But actually, I don't think it's typically true. So you always manage to fit something in your timeline. And if the motivation for me was to generate this company, I had the motivation. I found the time, right? It is effort. It's a lot of time. It's a lot of work involved. But I think time typically is not the limiting factor. It's more oneself the limiting factor. So you need to change your routine. You need to, do shorter. I don't know. We never had that TV at home, so I didn't spend time in watching TV. Let's put it that way. But somebody that used to have every night 2 hours TV time, maybe you will need to cut that out. But this is already a difficult, because you have a routine and you think that, no, this is my time. I want to have it, and so it's a matter of prioritizing. And I think the time you find it, it's not typically the problem. I grew in a generation maybe that this work-life balance was not so present. But I don't think I miss a lot of my life. So I still believe I work hard, but I also have a good time with my family, my hobbies, and social contacts. So I think it was intense. But I think this is exactly the period of time where one can be intense, right? When you are between, I don't know, 25 and 40, I would say, you can really put intense. And this is a time where you need to push to your dreams and find things that... So I think this is really, I don't think it's a matter of time. Time is there. You just need to adjust your schedule and your routine to find the time. But you need the motivation. If you don't have the motivation, I guess that's what it gets against you.
So motivation, inner drive, and then prioritizing so your schedule, your tasks. And what was, did you have a wife, I mean, at that time? Life, of course, you had.
Yes, so I got married the same week I got my PhD. So in that week I got 30. I kissed Ganzeliesel, which is the classical tradition here in Göttingen, and I got married in the same week.
Wow. This is a lot of memories.
So it was also very intense because I needed to organize the wedding together with my parents coming for the Ganzeliesel ceremony. It was a very intense period, yes.
So, but your wife is supporting you in this activity.
Yes,
this is
exactly.
very important.
This is you need. Important. I mean, without support, is you cannot get it much, especially if you are wanting to have a family. Yes, yes. Wife has been super. My wife has been super supportive the whole time, and I think she understood at that period that it's needed to spend a lot of time in front of a computer or moving around. Of course, I can many times say it would be nice that this weekend you spend less time in front of the screen, but and it happened, and I think we're enjoying. I think we got a balance in there.
Yeah, I also don't like, to be honest, this work-life balance, because when you work you don't live, or what is the case? I mean,
exactly,
we choose our job or our careers so that we love what we're doing, and we go there with passion and not like...
At least in ideal case, right? So exactly, this was... I was so inside my idea of developing that and moving forward with this, that it didn't feel like a job. Didn't feel like it was investing a lot of time, but it was not a time that I didn't want to spend. It was actually enjoying what I was spending. And I think when you find something you really like or motivated in your work, it doesn't feel like a load or difficulty.
Exactly, this is a cliche, but I also believe in that, and I feel that way when I go to work, or when we do this, for example. I feel joy, and it's a pleasure to do it. And I don't feel like I have this burden of, okay, there is a lot of stress, I cannot cope with it. No, it's like relaxation time sometimes to go. Our job is basically to discover new things and put the knowledge that is on the frontiers of humankind, and it's very exciting to be part of it, at least to me.
But this required a lot of commitment, a lot of time investment, and which it doesn't feel wrong, because you're enjoying it, and then it's not a problem.
Exactly. I completely agree with you on this. So now you have this company, and which is called NanoTag, by the way.
Yes,
let's mention the name. Exactly. And it is located in Göttingen, but you also run a lab in one of the institutes here.
Yes.
Can you please tell us more about what is your role as a PI in your lab, and what is your role in the company, for example?
Well, the role as a PI is quite clear, I guess, for most of the people hearing, because they know what a group leader is supposed to do. So it's basically having a bunch of projects and guiding, mentoring students, basically solving problems the whole day. So this is always coming problems that they need some solution, and of the projects trying to get money, fundings from the next projects, and writing papers. I think that would be the life of a PI. I try to do still experiments. Whenever I can, I do experiments, but everyone around gets annoyed because I don't know where the pipettes are or where the things should be. But I still like to do experiments, so I whenever I can, I do. So that's a group leader and responsible of people trying to get the next level of education at a PhD level or doctoral title, which I think, in my opinion, is where people should learn how to get independent, how to plan experiments, how to be critical about the results and know that this is an artifact or not an artifact, what controls are missing. And I think this is what is the learning process at the PhD level. Unfortunately, there is also a lot of things to learn on the side of this because there is new technologies, and then you need to get into this and making to work. Or sometimes you put there are ideas of doing experiments where the lab doesn't have strong expertise, and that means you need to establish things from scratch, and this is actually tough. On the other hand, interesting or fun, I would say. So I think this is a PI, and in the company, I used to be the CEO. So for a couple of years, not at the very beginning. Then it was a transition where I was the CEO, and now also I give it back the CEO. So I retract a little bit of this decision making in the company, but I'm very much into with what's happening with the drive, where the company should go, what are the type of things we want to create, what we want to build, how we want to make better tools for science, and this is still the motto in my view of the company. So now I'm more in the technology advisor in the company, but I'm still very much involved. I spend quite some hours a week there as well.
So you already found out your routine between...
At some point it was very complicated because I was the CEO of the company plus the PI, and this was really taking a toll in terms of time and difficulty to get. And unfortunately, you were not doing both of the jobs. You were not doing it fully correct, because you were missing some hours to do these things properly.
That is true. But what is the skills, for example, and knowledge that one should have for a PhD in your lab, for example, and for a work for like scientists in the company? Is there any difference, or do you need the same skills to do both? What do you think about this?
It's true for basically most of the biotech companies. You need to know your science. You need to be precise. You need to... Company science, I like it very much. I learned to like it. So this was something, one of the first meetings, company-oriented meetings, where only companies come basically to do B2B, this business to business, and try to cut deals and everything, and talking about science. They were talking about science in meetings, but it's a very different meeting than a symposium, a scientific symposium. I was shocked to find out that actually the business world, the science business world, is not trusting the academic business world. So they tend to reproduce, if they are interested in a paper published in university something, they, before moving ahead of doing something there, they try to reproduce first the paper, only then will think if this is true or not. So I think there's this reproducibility issue, which is unfortunately hitting very much the academic system. In my opinion, a little bit of how things are being, the policies around and how things are handled, what are the next steps, and the speed that science is being pushed to have, which I think is complicated to force results to happen in certain period of time. Well, companies, they decide very fast. If things don't work, they drop the project completely. And after they have invested millions of euros, after a year, if things that they are coming again and again, not moving ahead as they expected to move, they just close this project. This is very hard to see in academia, unfortunately. So people always try to rescue their project, because you had these 3 years where you need to invest, you need to get a paper. Of course, might not be a Nature paper, might be a smaller paper, but you always try to rescue it. I understand both worlds, but I think it's a little bit different how science happen. I do like the science in companies because they need to deliver things that has to work, that they will be validated for many others, and they will be under heavy control. So they don't have the luxury of publish a result that the controls were halfway correct, which in academia happens.
Yeah, and this is actually counterintuitive in terms of the thinking of the public related to science done in the company and science done in an institution or university. People tend to believe that actually universities have this more truthful way of doing science than the companies, but maybe in reality it can be the other way around. So as you said, they're very much regulated, and the scientists are not that regulated, and you need to publish a paper in order to get a nice funding and continue your other project. But how would you think, you mentioned this reproducibility crisis, which means that some papers, so the work of a scientist, as you know, is to write publications about their research, and many times in this in some scientific domains, as in neuroscience, people around the world in other labs, they are not able to reproduce certain experiments, and there is this so-called reproducibility crisis, which kind of prevents science to progress the way it should be, in my opinion. And what do you think is the problem there? You already mentioned this kind of need of a funding, but is there any kind of solution that you thought about it during the years?
Funny enough, one of the molecules guilty of reproducibility issues are antibodies, right? So, and this is also what I'm very happy about our nanobody work, because antibodies, if you would produce them in a rabbit or in a goat, they are typically polyclonal. We did not discuss that. But that means that these particular antibodies have a specific features which only come from that particular rabbit or that particular goat. As soon as this animal is not there anymore, then this antibody doesn't exist anymore. And so work that has been done with that particular antibody, it cannot be reproduced in the future because this antibody does not exist anymore. And this generates problems. The next problem will be the poorly validated antibody or affinity probes in general, where they bind only not a target that you wanted, but it binds also something else. Or it was never tested for that application and suddenly give you an artifact results. It's a very laborious and painful process to validate a molecule in a very correct fashion until the end. You cannot for every system. So I cannot predict that this antibody will behave exactly as I did it or validated in my neuro cultures, if they will behave the same in a B cell or a T cell culture, right? And they might start binding to proteins that in the neurons are not present. But you will need to validate for every tissue, for every protein, for every species different. Because again, if I do it in monkey, or if I do it in an experiment, the staining, I do it in a mouse, or if I do it in a organoid or human-derived cells, it might behave completely different. It's a complicated question. So, and I think most of the problems in reproducibility a lot of them comes from antibodies. So why nanobodies? Again, I think we are helping, at least is one of our slogan, is to that we help this, is because nanobodies are recombinantly produced. So once you know the sequence, you can produce them to infinitum, for always, with the same features. So if I discover something with this nanobody today, it will be the same nanobody in 200 years from now that will be produced. And I hope that if somebody wants to reproduce my science with this nanobody, it should be able to reproduce it because it's exactly the same antibody. It's recombinant. It doesn't depend on an animal anymore.
This is also a very nice feature of the nanobodies.
I also think so, yes.
And for the people who don't know, the sequence is basically the code that the cell used to produce this particular protein, or in this case, the nanobody. But you mentioned, we talk about these nanobodies, antibodies, but I think you also have other products at your company which you have developed in recent years. For example, a very nice one that we use in our lab is the so-called AlphaTag. Could you please mention more about this invention?
Yes, so we are also very happy with this. So it's again generating tools. So we like to make all the it's kind of a Lego situation where you build stuff. And this is again a nanobody binding to a very small domain of a protein, which is called an alpha helix. It's only a 13 amino acids, which is really, really, really small. And this nanobody alpha, what we have binding to it, it binds with a very high affinity and a very, very high specificity. So this was designed. We designed this in order to have several features, which is a lot of details, which I don't know if I want to go into details, but into deep on them. But it has become a very important tool, and I'm very happy. This is something that a discovery that it has the implications of this AlphaTag has been for my research difficult to achieve in the academia, because I get a great paper in academia which is cited 100 times, and I'm happy about it. Because my contribution to science have helped another 100 papers to actually move further. Well, this has been exponential with the alpha system. So we put that thing out, and we are seeing how people is using this tool in diverse fashion in all type of fields, from plant to therapies to diagnostics. Name it, and this is very rewarding to see that a tool that you created is now being used all over the world in different applications. I think this is I haven't had this with a paper, I have to say.
That is true. It should be very satisfying, and we all actually hope as young scientists that we will produce something out of our research that will be used around the world. Yes. Often this is not the case. We build science is like a house of cards where you put a little bit. Yes. Right now, and because of this reproducibility or many other problems, maybe some things will collapse. We hope not. But as a young scientist and people who are below us, also we would like to kind of contribute to the changing of this situation right now. Mm-hmm. But speaking of application, so we talked about the technical side and the nanobodies and the company, but how do you use these things, these tools in your scientific career and in studying the brain, for example? How can you use these guys and have more insights about the function or structure of the brain?
Mm-hmm. So yeah, the aim at the academic side is to generate enough nanobodies recognizing different players in the synaptic transmission, and either at the pre-synapse as much as the post-synapse. Traditionally, the lab has been focused way more in the pre-synapse, but I think the post-synapse is equally interesting. And this is an amazing structure because we have trillions of them in our brains, in our central nervous system, and we're still not fully understanding how they communicate, all the molecular processes, how neurotransmitters are released, what are the machineries. And this has been a lot of effort, amazing works around. But as I said, the new tools like super resolution and other now cryo-EM and so forth, I think they are really is an exciting time, because they are to a point that we can see things that 10 years ago was impossible to observe. And I still believe that for most of these observations, we still need a specific way of tagging them, to label them, that I know that this is protein X and this one is protein Z, and this one is protein W. And for that, I still believe nanobodies are playing a crucial role for this very small size. They are very accurate in labeling their targets. So yes, my lab focus on understanding some of these mechanisms. So one could think a super resolution to look at the nano anatomy of a synapse. That's what we aim, and ideally not to a single protein at a time, because then you see a couple of dots in the void. I would like to see them in a context, and that's why it requires that you don't tag only a single target, but you tag ideally 5, 10. Today 3 still possible, difficult but possible. We would like to have more. I think this is one of our aims in the academic lab. And the second point that I really like to exploit out of the nanobodies is that, different than antibodies, nanobodies can be produced recombinantly inside the neurons, and they can bind their targets when the neurons is alive. So you can really use this as sensors or detectors or biosensors. So creating biosensors is also, that for me, is a very attractive way of using them. So to make a nanobody bind to a particular target only if something happened, or basically to stop a process, to inhibit one of the mechanisms, and this will help very much to understand what are the necessary steps for a neuron to release neurotransmitter, for example.
Okay, but if we go a little bit technical on that particular point, when you express this nanobody in a neuron, would this nanobody have some fluorescence capabilities, and also would you actually perturb somehow the function of this cell, or nothing will happen?
This is a risk for everything you investigate. As soon as you start looking to a system, you're modifying the system by any means, by applying light, by having it at different temperature, by extracting it from its tissue to a single cell. So you always do affect. That is true. And this is important to, again, rigorously check that the nanobody, or the what we call intrabodies now, do not affect the conventional physiology of the neuron. It needs to be checked. I mean, this is for different nanobodies, you might have an effect. Sometimes you want to have an effect. You want that, for example, blocking a phosphorylation domain, and basically you can now with optogenetics and other tools that are appearing, you can manipulate nanobodies to turn on and turn off inside the cell by light or pharmacologically, and then you are able to let's say block synaptic activity at will by turning a light at that particular synapse without blocking the other buttons, for example. So in terms of understanding networks, that will be also a very, very interesting approach to be able to manipulate or control neurons at that level with that precision. But it's complicated, right? It's always demanding task to generate such a molecule that is not affecting physiology, but is, I don't know, giving you some ideas of where that protein is at that time. Yes, fluorescence is what we use, but it can be used other things. There are isotopes you can use. Then for live mass spectrometry will be problematic, cryo-EM as well. But that's why we like fluorescence, because we can follow this in living neuron. Unfortunately, shining light, collecting light, so the cell always will feel not as that in the brain.
Yeah, but as we know, we use microscopy, light microscopy in all of our experiments, and we use light. We cannot gain the information without using light and exciting this fluorophore. So maybe I think after some controls you show that actually these guys are not perturbing the physiology that much, and this would be a nice tool that we can use in our research, right? And but these are intracellularly expressed. But you mentioned also, yes, we have a limitation in terms of how many different colors we can use to stain or our cell, for example. Let's say in our case are maybe 4, but you would like to have even more colors. How is this possible? Would this be actually applicable?
This is an active area of research in my lab. So increase the multiplexability of microscopy. So the idea will be not to only look at 3-4 targets, which is the conventional way of doing light microscopy, by separating the targets in different spectrum in the chromatic spectrum. So you will just give to protein A color blue, and you will give to protein B color green, and so forth. But effectively, you can separate very well 4 colors, maybe a little bit more, but actually let's take it to 4 colors. That's a possibility. But now, again, there are appearing new sensors for lifetime, for example, where you look at the decay of photons upon a fluorophore. So a fluorophore that gets excited, it has a particular characteristic of giving photons, and you can trace that in time. So you need to have a very fast detectors that they are detecting the time where the photons are coming, and then you can start separating two fluorophores that they are from the same spectrum, let's say green, but they have a different way of giving you the photons, and you can start understanding this. So already with Lifetime's new detectors opens a new possibility that you can look at 5, 6, I don't know, we are dreaming with 10 different targets in the same sample. So in the same synapse, I will be able to detect and segment 10 different proteins. And this is what we are aiming to. I mean, even more with other techniques, with currently with exchange paint, which I have not talked about it, but is another type of super resolution microscopy, where now every nanobody doesn't bring a color, but it has a DNA sequence attached to it, and then you have a complementary oligonucleotide only for that particular sequence that it will tell you about protein A. Another sequence of DNA will reveal you the protein B, and another protein of another sequence of DNA will reveal you protein C, and so forth. And this is a very, very flexible tool that you can actually theoretically go to as many colors as you have nanobodies.
That is actually mind-blowing. Can you do this also in vivo, in a living cell, or still on the
lifetime might be possible to do it in vivo? Actually, it is possible. It's because the photons are much fast. The exchange paint technique is more complicated in vivo, because for every color you need to acquire 10 to 30,000 images, and that make it that it gets complicated for life when things are moving. Doesn't help.
As we know in a microscopy. Yeah, exactly. But these are exciting projects, and I hope your students, which we well know, get to a point where they develop some these tools further even, and we can all benefit from them. But what are the nanobodies that you would recommend us for studying the presynaptic and postsynaptic side, for example? Which are the best in your opinion?
So, specific nanobodies. So the specific nanobodies that now are currently nicely validated, working very nice. We have against a calcium sensor, which is a vital protein. We know that upon calcium entry in the presynapse, synaptic vesicles decide to release their content, the neurotransmitter, and talking to the postsynapse. And this calcium sensor is called synaptotacmin, and we have a pretty nice nanobodies binding to that protein. So I hope that this nanobody, with the precision we can have, and also works as an intrabody, it can give us nice insight of how this molecule is working in helping synaptic vesicles to get fused to the plasma membrane and release their content. That will be a presynaptic nanobody I like very much. The second one is one that is coming from NanoTag, actually. It's against the postsynaptic density 95. In short, it's called PSD95. It's a protein that is a scaffold protein found in the postsynapse in a region that is called postsynaptic density. The name comes because even by electron microscope you could see that region. You can definitely recognize the high density of proteins compared to the presynapse. So in an electron microscope, although you don't have colors, you were able to immediately recognize what is pre, what is post, due to this shadow or high density of generated by this scaffold. And one of the protein there is PSD95, which is you find it in all excitatory synapses. So only excitatory synapse will have PSD95, and this is the common belief, and we have a nanobody binding there. So the nanobody there has a major advantage because size again. So antibodies were struggling to label this PSD95 due to the high crowding effect of so many protein in that region. It's like a gel. It's so many protein. But now nanobodies, since are much smaller, they find their way, and they are able to reveal you in a much nicer way the postsynaptic densities. And I think that I would say are the two nanobodies I like them very much for the study of pre and post synapses.
Exactly. So synaptotagmin 1 for pre-synapse and PSD95 for post-synapse.
Exactly.
And this is actually another feature that I like about nanobodies, that sometimes you have these epitopes that you cannot access with the antibodies, the conventional ones, and you need smaller molecules to get there and give you a signal from a particular epitope or a molecule of interest. And this is another. And also the third feature, I think, is this so the decrowding, and you reduce the quenching, self-quenching of the fluorophores that sit on the antibodies, right? So because of their size, which is just astonishing. Yeah. But okay, so we talked about your path as a scientist and some problems in science, and also about the company and the very nice nanobodies that you have and the problems that you want to address. But what is actually the one problem that you would like to tackle in your lifetime as a PI and say, okay, I want to solve this particular neuroscientific problem and be happy with it after my career? Is there such a problem?
I don't know if I have such a unique goal. I think there are several, and as my career was showing, I tend to jump very often from topics or fields, and so I think I would stay definitely in neuroscience, and I would like to contribute more to neuroscience. But do I have such a defined one? I don't know if I can answer that. So yeah, clearly for me, it's to understanding the synaptic machinery. Also at the postsynapse, I think is very interesting. And I think slowly we're having the tools. I don't know if I can compromise myself on saying this is the goal I want to go.
Yeah. So maybe on the way, during your further challenges, you can maybe discover another one and say, okay, this is the new target, right? So great. I would love also to ask you another thing. We are slowly progressing in time, but we already mentioned about these problems in science, for example, in terms of reproducibility. However, there are other problems like the publishing process, the review, or the grants that we are getting. Also, there is some kind of a crisis that we know in the community about the postdoc crisis, that many people after their PhD decide to go to industry instead of going to academia. Could you address one of these problems? We already talked about reproducibility, maybe something else, and then tell us how would you like this to be solved in your opinion, or is there something to be done as for us as a young scientist, for example?
Yes, so I think the one that I would like to address maybe is the tendency of scientists to now go into industry, and I think for that the academia needs a renovation, a refurbishing of how things are running. And somehow I think the academia should not try to even compete to the industry. They should try to find different niche of what is interesting, and it should be the case where in academia we thrive for knowledge, and where in the industry it's very important to generate a final product that you can commercialize. So I think that is something that the academia should stress even further to research on basic questions to understanding the roots of things. Not necessarily that will have a economical implication in 2 years. And saying that, I was reading just recently a little perspective in Nature, which I like it very much because it was good science take time, and it's today the policies are all to you get grant to hire a PhD for 3 years, and you need to make it in 3 years, right? So in these 3 years, the PhD need to have learned everything, generate an amazing amount of data, and also the high quality data, and making a breakthrough in science. I think this type of policy pressures are complicated because it's hard to pressure scientific discoveries and development, and I think they take time, and take typically more time than these pre-designed periods. So this is one aspect that it suddenly becomes for many students or many new people in science. They are not secure perspectives in the academia, and this also make it more attractive to jump into the industry. It's also not a super secure, but at least typically salaries are a bit higher. Stress level is a bit lower. So I understand that it can become more attractive to move into a big pharma or something like this. Which is it's okay, but I think, and I don't see it as a problem. I see it as an opportunity for the academia to find a new niche, to find a new way of doing science, and maybe change the science. That now we don't need to put in grants that we are going to cure Alzheimer and cardiovascular diseases or solve the climate change in order to get funded. We should find the basic reasons, the fundamental reasons of things. I think that's where academia should be mainly focusing, more than competing with the pharma in a way.
That is indeed an interesting perspective, and I tend to agree with it in terms of to clearly define that we are doing long-lasting projects that might not be turning that profitable, but indeed they contribute to the progress of the scientific work. Without these small steps, we will never go further, right? Exactly. This is, I agree with you to that point.
I see why it's complicated, because you as a PhD student also realize that at some point you want to give it a cut at this period and move ahead with the next steps, okay? So it cannot be also without any limits. But some of the very strong limits that now policies are making, I think, make it complicated to generate high quality, reproducible science, because in that relatively short period of time, you need to generate a paper and move forward. And this is where I think some of the non-reproducible papers might come up.
Exactly. So we have already connection between two problems: how science is being done and also the reproducibility. This is actually true. So I think we are going right on time with our conversation, and I'm really happy that we had you here during this roughly 1.5 hours, and we discussed a little bit of your path, as I said, as a scientist, a little bit of a career in terms of industry or a scientific career, some problems in science, and also a lot of technicalities that I might, and our colleagues that are behind these cameras, clarify for you guys out there who would like to know more about. But really, I enjoyed this talk with you, Felipe, and thank you for coming once again. And we were happy to have you, and of course soon we're going to release this episode and we will share with you everything that you would need to share it also. Exactly.
Thank you very much from my side as well. So it was a pleasure, and I hope this initiative of interviewing scientists really grows and get successful. I like the idea very much.
Yeah. Thank you. neuroscience and beyond. Know more, get inspired.